Okay, and like always, I just started the YouTube recording. So if you, I don't know, want to change your name, now's the time. Okay, so we'll be starting with today's lecture. Today's lecture, we're just doing bio and biochemistry practice passages, right? Just like we did this whole week with different topics. Again, this is the consent to audio and video recording. This is my personal introduction. Again, I highly recommend the GroupMe if you're not already part of it. Moving on to the content. Since today is actually the last lecture of the series, unfortunately, but we'll be doing something else. We're going to be taking a quick break for a couple weeks and then we'll be moving on to something else, but you'll hear more announcements about that coming up. Right. So first, jumping right into it, we're going to do biology practice. Last time I used Jack Weston. This time I wanted to use this resource. I'm doing this just because I wanted to introduce some free resources that you guys may want to use as well. When you're doing your practice, just so you get an idea of what's out there, what each program offers. Out of the free resources, there's also a lot of paid ones that have practice passages as well, obviously, right? So the link to this passage is also in the Google Drive that's linked in the chat, or you can follow along here. Graphs may be a little bit difficult to see, but I tried my best to make them visible and fit everything on here. All right, take a couple minutes. right, please take another minute or so to finish up. And by the way, again, if you guys want to just message me when you're finished, so I can know. Okay. Okay. So moving on to the question, and I always encourage students to go back to the passage after they see the questions, just because you don't really know exactly what you're looking for. Some questions may not need it, like for this one, you probably won't need to go back to the passage that much, but for some, it may be very helpful, right? Take a minute or so to answer this. It shouldn't take you that much longer. Then if you want, message me your answer choice. Okay. So if everyone's ready. So the correct answer is actually a gap junction. Oh, sorry. So yeah, this is just a simple knowledge question, right? Gap junctions, because it mentioned CX43, kind of junction to CX43 form, mentioned its connexin, mediates direct transmission between neighboring cells. So in this case, it would be gap junction. Desmosome, hemidesmosome, I don't think I've ever heard these words ever like shown on MCAT before, so I highly doubt that you'll see them in a real MCAT. Tight junctions, tight junctions are a little bit different than gap junctions. Won't go too deep into the explanation because they're not really also that common, but just know that when you use gap junctions, which type of cells use gap junctions, which type of cells use tight junctions, and the difference between them, okay? But like I said, it's not really too common. Next question. Okay, this one's a little bit more difficult, but still pretty straightforward. Again, doesn't really involve too much looking at the passage, so we'll give you guys a minute for this one. Okay, so the correct answer is actually C. The inhibitor diffuses through the cell membrane. What do you know about this inhibitor? Well, it's made of a glycerol backbone, 3 saturated fatty acid tails. What does that sound like? That sounds a lot like a triglyceride. Well, because glycerol and 3 fatty acids sounds a lot like triglyceride. What is a triglyceride? That's a lipid. How do lipids usually get in cells? Well, what are some common lipids that are found in our bodies that we usually use? Well, one of them could be hormones like testosterone, cortisol, things like that. Those are common lipids or similar molecules. And how do they get into the cell? They just go right through the membrane because, again, the membrane inside the membrane is very hydrophobic. So something hydrophobic, like a fat triglyceride or hormone molecule, can just go right through, right? So that's why it's C. The inhibitor is recognized A and B. The inhibitor is taken up by a non-specific receptor or by a specific receptor. That would be more like a peptide molecule or something that's not some kind of signaling molecule, or something that's not necessarily like similar to a hormone or something that is like a signaling molecule more than like a lipid or fatty acid. And D, the inhibitor passes through the cell membrane via channel. Mainly, the only things that pass through channels are ions. So this is not an ion, so it wouldn't be D. Okay. And this is the next question. This one's a little bit trickier, but please give it a try. Okay, take another 30 seconds to a minute to answer this question. And for the new person that joined us, we're just going over a question. We're doing practice patches today, just like we were this week. Yeah, and here the passages, me and Chris just linked them in the chat to the Google Drive. Okay. So this one's a little bit tricky, so I'm going to spend some time going over it. The correct answer is A. Right? And let me explain why. I think for me, when I was reading through this question earlier today, I thought of it during using process of elimination. So we're deciding to interrupt this interaction, right, without using a small molecule inhibitor, which would most likely disrupt this interaction. Well, plasmid producing this would obviously not work because it would make just more of the CX43. So that wouldn't really make sense. Acetyltransferase specific for B-actin. Well, B-actin is mentioned, and I'll go back to it in a second. The passage of B-actin is not really mentioned at all in the passage. B-actin was used as kind of a housekeeping molecule for one of the tests. I think that was the only time it was mentioned, but otherwise it's not really relevant at all. And radiation against heptanol, that's not exactly relevant either, so wouldn't be this. And so the correct answer, just by process of elimination, could be A, but the reason is because siRNAs are specifically can be used for doing this. And here, I'll go back to the passage. Right. So like I mentioned, I think this is the only time they mentioned B-actin, and if you notice here, it's just really just like a housekeeping gene, or it's just something that you know that the proteins that the cell is going to be expressing. So it's kind of like a control group. Should be expressed in all three, and it's just kind of like to show the difference between 43 between the three groups. And then I believe they mentioned strategies. Right, they used heptanol, a well-known inhibitor, to alter the function of CX43. So that's why radiation against heptanol wouldn't really work, because it's the inhibitor, so it doesn't really make sense. Why B would be the right answer, right? And they confirmed the interaction through yeah, and really it was just by using process elimination. That's the main way, unless you know what siRNA is, right? Which is very rare, so I would expect most people wouldn't. In case you do, it was even simpler, but really you could do process elimination. Just to, this one doesn't make sense, this one doesn't make sense, this one doesn't make sense, so must be A. Okay. Okay. I take, this one's a little more complicated, and this one's based on the passage, right? So the first couple were based on outside knowledge mostly, or just looking at the question. This is mostly looking at the passage now. So take a minute or so with this one. Okay, if you guys are ready to go over it. Okay, the correct answer is, for this one, is actually C. And let's go over why. So A and B, all expression control occurs at the transcriptional or translational level. First thing you have to recognize, transcriptional means the amount of mRNA will be different. So the actual transcription of DNA to RNA, and translational means RNA to proteins. That at that stage, the way we can actually look at this from this graph is we can see that this is actually measuring the mRNA levels, and this is measuring this is measuring the mRNA levels. The graph is measuring the mRNA levels, and this is measuring the protein levels. These black bars. Okay, so we noticed that if it was just transcriptional control, then we would see it as we do here. In here to here would be there'd be a big difference, right? Because if it's transcriptional control, it gets changed at the mRNA level. If it was translational control, it gets changed at the protein level. So the amount of RNA is actually should be the same. So if it was at translational control, it would be this would be exactly the way it is, except these three bar graphs would be about at the same level, right? And only this would be different, really. Okay. If it was all transcriptional control, then this bar graph would be the way it is, but this would actually not be this way. It would actually all be you wouldn't see that this effect, right? Because if it was transcriptionally controlled, you would have RNA, but then you wouldn't have the RNA to make the protein, right? Okay, that makes sense. So because you have both the bar graph different and this looking like this, between these two especially, right? They're the same. So it means that there's both transcriptional and translational control. So A and B are both wrong because it's a little bit of both. Okay. Now I'll maybe go over that again afterwards. Okay. But hopefully that makes sense. A and B can't be right because it's not all expression occurs at this level. The expression occurs at both levels. D, normal livers treated with heptanol have lower levels of CX43 compared to donor treated with heptanol. Well, it just says based on figure 1. So we're really just looking at figure 1. Normals compared to donors treated with heptanol, we're just looking at this graph. We don't really kind of see that at all. So it wouldn't really make sense for D, right? C is much easier to see. Donor livers have high levels after reperfusion compared to normal liver tissue. This is normal liver tissue, right? This whole column. Actually, I'll do this. This is normal liver tissue, and treated with have higher levels of CX after reperfusion compared to normal liver tissue. This is after reperfusion, right? You can see the amount of mRNA is higher, amount of protein because of this band is higher. It's very simple, very easy just to see that. So see the correct answer, B, A, or not. Heptanol we're not really talking about right now. Not really. We're talking about reperfusion, and we're just basing it off figure 1 because that's what they told us to do. Okay. And again, expression occurs at the translation versus transcriptional level. It can be at the translational level only because you can see there's difference in mRNA. It can be at the transcriptional level only because even though you have differences here between this and this, these are equally as, because there's a difference between the mRNA levels, this and this has no difference, right? So because of that, there must also be some transcriptional control as well. Otherwise, these would be slightly different. These two graphs here, these two lines, how would they called? Sorry, bands. These two bands. Okay, hopefully that makes sense. Before we move on to the next practice passage, any questions about this practice passage or any of the topics I've covered so far?
Yes. Sure. I have a question with the. And now is this, yeah, the first question with the connexins and desmosomes. Could you go back to that one, please, Mark? real quick.
Yeah, sure.
Yeah, I know most. Oh,
Yeah, could you
I know
just
I
mute
know most
yourself?
pre-meds probably wouldn't have advanced knowledge of having desmosomes and desmosomes, but you said just have a general idea. But I mean, those desmosomes, they actually connect cells. So when I see connexin, I'm thinking that there's like transferring, because the passage did mention that it allows neighboring cells to be affected. So I think desmosomes does that also. So how would I not? I mean, I got it right because of gap junction, but desmosomes kind of allow neighboring cells to influence each other.
Right. So to be honest, like you mentioned, I actually don't remember. I might have learned it like at some point, but I really don't remember what these are. So to be honest, but I know that gap junctions would be the best option here. So I just went with that right away, just because gap junctions are the things that connect cells like directly, and as they were described in the passage, that's kind of like the best way the cells are connected. Seems to be the right way. Tight junctions are a little bit different. You can do some research in tight junctions. Desmosomes, hemidesmosomes, I really don't know that much about. I could do some research and get back to you, but like I said, I don't really remember that. And I don't think, just because it's like, I've never heard that before on an MCAT. I haven't seen that in a really long time, since maybe.
Okay, we can move on. Like you said, gap junctions is the more familiar term. And in terms of function, it did what the passage described.
Yeah, exactly. All right. Any other questions from you or anyone else? Okay. If there are no questions about this, we can actually move on to our biochemistry practice passage. Again, the link is right here, but I'll be posting it along, but I'll also post it again in the Google Doc here if you want to follow along on your computer. I think it's easier a little bit to see. Like I mentioned earlier a little bit, but I want to reiterate again, I'm using this site today. Last time I did it, I'd used Jack Weston. I just want to show you guys all of the free resources that have practice passages that you can utilize when you're studying. There's also a lot of paid ones, but I wanted to showcase some of the free ones that you may easily have access to. So this is one of them. Jack Weston is another. Okay, if you guys didn't know. Okay. Take about, I would say, five to 10 minutes. This one's a little bit more complicated and a little more in depth, but still, again, try and keep pace for your timing. About 5 to 7 minutes for this passage. Okay. Okay, I'll give you guys another minute. I know this one's a little bit denser and a little bit like, especially the graphs, a little bit hard to look at, but I think you'll be coming back to it a lot more for the question. So I would just say, if you get as much information as you can in the next like 30 seconds, then we'll move on. Okay. Okay, let's move on to the first question. Like I said, I feel like you'll be coming back to the passage a lot for this one, and especially to the graphs and just everything the passage, right? And I'll give you guys a little bit longer for these questions because I think they're a lot more in depth. So take maybe a minute and a half. Okay, let's go over this one. Okay, so the correct answer is B. Okay, which the following is the most reasonable conclusion based on figure 3, right? Wild type cells lack adequate levels of ER phospholipids. That's not really supported by anything here. So as you can see, wild type is this dark red. They do have much lower levels of phospholipids than these, but it doesn't mean that they lack adequate levels. In fact, they probably have normal levels. It's just that the deletion mutation actually has high levels rather than adequate levels. So A is not really supported. We don't really have information for that. C, this Sec59-1 acts as a transcription factor to repress lipid expression. That could be one reason, yeah. We can see that when in the wild type, the levels across all of the phospholipids at all time frames are lower than those of the deletion mutation. If you deleted Sec59 and the level of phospholipids went up, one plausible explanation is that it's a transcription factor that represses it. Okay, that makes sense. Sec59 is the primary repressor of phospholipid overproduction. Again, that it represses 59 phospholipid overproduction, which again, because you know that once you get rid of it, you have overproduction. So yeah, that's a plausible explanation, maybe. But for both of these, yeah, it's a plausible explanation, but you don't know that. That's one possible answer. And you have to do way more testing to find out. Again, it's the primary repressor of phospholipid overproduction. Well, how do you know there's not others? How do you know it even is a repressor of phospholipid overproduction? You don't know enough information to say it's B. Same thing for C. Sec59 acts as a transcription factor to repress it. You don't know that it's a transcription factor. You don't know its mechanism of action. You don't know anything about it. You just know that when you get rid of it, you have more. But you don't know how, you don't know why, you don't know anything like that. Just based on this graph. So while C and D are plausible, they're not exactly the right answer. Whereas B, cells lacking Sec59-1 contain higher levels of phospholipids compared to wild type. Very simple, very plain, very easy, just super basic, and that's what you get, right? You don't have to do a lot of thinking. You don't have to make a lot of conclusions. You don't have to make a lot of inferences. It's just based on what you see on the graph, and you just report what you see on the graph, right? What you see on the graph is you have wild type, you have small amounts of phospholipids, you get rid of the Sec59-1, you have a lot of phospholipids, and that goes for all the phospholipids here. Okay, very simple. So you lack 59, you have high levels of phospholipids. Does that make sense to everybody? Why C and D are not right, and why B is just the simpler and better answer. Okay. So I'll move on. If not, please put a question in the chat or feel free to speak up. Okay. Now we'll move on to the next question. Again, this is another graphing question. So okay. Everyone's ready? Correct answer is C. I think the biggest hurdle with this question is really finding out there's like so many colors, so many bars, so many everything. What does everything mean? So I think you really have to take a little bit of time to just kind of like find out what everything actually means. All right. So this first burgundy, I guess, color is just a wild type. This is just the normal amounts, right? And y-axis again is amount of phospholipid. The x-axis shows which specific type of phospholipid, but really the trends are basically the same throughout for all phospholipids, so the x-axis doesn't matter too much. The y-axis is just the amount, and the colors are really what's important because they're measuring the different variables. This first burgundy color, the leftmost one, is just the wild type is just what's normally there. Okay. The green is the wild type plus this YEP352 gene, right? And this was what kind of we're looking at, right? And that's kind of like, that's the plasmid, right? So if you have that plasmid, that's what Sec59 overexpression is, right? And I'll go actually back to the passage to make this, to show this even easier. Then overexpression might also have an effect, right? To overexpress Sec59, researchers clone the gene into this vector, right? You guys know what a vector is? It's kind of like a plasmid or something that you, for genetic engineering, you just put it in, and then you into this genome, bacteria, virus, sometimes it's different, whatever, and then you just put it into cell you're looking at, and it'll kind of repopulate essentially, right? It's a way to just put a gene into something. And the gene that they're putting in, specifically in this case, is the Sec59 gene to overexpress it, okay? So having this wild type plus this YE whatever is just basically means overexpressing Sec59. That's exactly what this green graph means. It's the amount of phospholipids that are present when we overexpress Sec59. Then this next graph, Sec59 with this I delta, right? And you may notice it here. This is the deletion mutation. So this is what happens when you don't have Sec59 at all, okay? And this, in this last yellow graph, is what happens when you don't have Sec59 normally, when you delete it from your normal human person, but, or animal, I guess in this case, but you also put in this vector, which normally would make it overexpress Sec59, but now it just normally expresses Sec59 because now there's only one copy again. It's not technically an overexpression anymore, right? So now that we got that out of the way, now let's look at the levels. You notice how the levels for this graph, and this graph are about the same, with this one being higher. We already knew that, and this is the deletion mutation. We know that the deletion mutation compared to the wild type was higher. We knew that from the previous graph we looked at, right? Levels of phospholipids when you delete something is higher, when you delete this mutation specifically, or this gene specifically, it's higher than the wild type. And again, if we look at all the phospholipids, these three graphs are the same, with this one being higher, all the same, this one being higher, again, all the same, this one being higher, so on and so on, right? So now, what is the question actually asking? What effect does overexpression have on phospholipid levels? We know that overexpression is this green graph versus normal levels is this burgundy graph. Just look at it, this burgundy graph, this green graph, they are the same level here, here, and so on. So phospholipid levels remain constant. They only increase in the case of this deletion mutation, which we covered in the last graph as well. They increase in the deletion, but in the overexpression, they actually don't decrease, right? So you'd assume if you delete it, overexpression, if you overexpress it, you would have less levels of phospholipids. That's not the case, as shown by this graph. And D is just like way too much of an assumption. You don't know anything about that, especially since phospholipids gluconeogenesis. You don't really, there's no evidence of that, and it's not shown in the graph, so this one just doesn't make any sense at all, right? It's really between these three choices, and out of those, C is the correct one, okay? And somebody mentioned the passage saying overexpression might also have an effect on lipid composition. Might have an effect. First of all, the effect could be increase or decrease, so it could be A or B, but it was might have an effect. That was before they did the experiment. This was the results of the experiment that's just how science works. Sometimes you think, well, maybe it does have an effect, and you realize, no, this has no effect. So might have an effect, not does have an effect. Opal?
Mark, we knew the orange one. Well, we knew from the passage that the wild type plus the YEP was the what you said, it was the vector. So we knew
It's the
that.
vector, yeah.
So that orange one, how did you interpret the what is that? What's going on? A deletion plus what's going on in the orange?
Right. So you know how deletions usually it's just like a knockout. Like you just take the animal genome. So we'll say we're working with mice. You just take the sec59 that the mice normally has, and you just delete it, or you either knock out, or maybe you can just inhibit it so it doesn't express at all, and it's essentially a knockout. But whatever, the mice's natural sec59 is canceled, but you add in this vector, which can actually add in the gene. Normally, when you add a vector into something, you're actually overexpressing it, right? Or sometimes you actually express something that's not there. So, for example, with the genetic engineering, they might have a vector that, if you have like some kind of default, like faulty genome or something that is not normal, they might add in a vector to give you some function that you're normally supposed to have, and that's what's happening in this case. Because the normal mice sec9 is gone, but they add in this vector into the DNA to make it basically have the gene.
Okay, so they knocked it out, but then they put it back. So it's functioning normally. Okay, all right.
Yeah, essentially, yeah. But it's not always like a 1-to-1 ratio, of course, because it doesn't like exactly always cancel out, because sometimes if, for example, you could have seen that this orange graph was still like this, right? That would have shown you that it even though they put in the vector, it might not be as functional as just if you had a normal wild type, right? Or if they had it like, I don't know, something else, then it would be different. But especially if they had it like this, like whereas this level, it would be closer to a knockout than it would be to the normal type. So it's not exactly.
Mark, the delta means that it was a deletion. Like you knew these what I said mnemonic, because did you know that the delta represented a deletion, or they told us in the passage? Oh, they did. No, the delta was the deleted.
Yeah.
Like 50. Okay, they. All right.
Right. So they told us that here, and mainly I actually looked at this because I knew that this graph was looking at the deletion only, right? The wild type versus deletion. I noticed that this is how they labeled the deletion, was just the delta, and I noticed it's the same nomenclature here. So I just kind of made that connection. But I think they might have used it here as well. Yeah, something like that. But yeah, you can use other graphs to help you sometimes. Like I use this. Okay, that makes sense. All right, next question. This one is a little bit more chemistry and physics based rather than biochemistry, I would say. So I won't give you guys too long for it, and especially since it's kind of like a general knowledge question. Remember, you will have a periodic table on the test. If you want, you could look it up. I don't think you really need it for this one, since there's only 2 elements, phosphorus or aluminum, I think that is. So, but just give it a try. Okay, take a minute. Okay. So, like I said, I don't want to take too much time with this question. It's not really that crucial, but correct answer is C. And the only thing you really had to know was what alpha decay means. Alpha decay is just a helium-4, right? A removal of helium-4. So what that means is you have, I'll use 32P as the starting. Basically means you started with whatever it is molecule started with. In this case, it was 32P. And then it decomposed or decayed into this 4 helium plus something else, right? And first thing you want to look at is the numbers, right? This is the mass, or yeah, the mass. They have to add up, obviously. You can't destroy mass. So 32 goes into 24 plus what? Obviously, then very simple, 28. So you know it has to have a mass of 28. Whatever the product is, it has to have a mass of 28. So this and this are gone. Down between these, now is it phosphorus or is it now aluminum? Well, remember that phosphorus has, I think it's 15, maybe 15 protons, right? It's not written here, but look at it in the periodic table. But really, you don't need to know this question. Just 15 protons. You know, helium has 2 protons, otherwise it wouldn't be helium. The number of protons determines what element it is. So that means 2 protons here. 15 goes into 2 plus how much? Again, you don't lose protons. So it would be 13. And I believe 13 is aluminum, which is C. You didn't have to know. You could have looked in the periodic table. You didn't have to. You just know that it would be another element, right? So the options are phosphorus, which is the same element you started with, or aluminum. Since you know you're losing protons, has to change element to a different element. So you know it's C and can't be A. If they gave you aluminum or carbon or something like that, then you would have to look in a periodic table which exactly it is to do the math. But in this case, they made it easy for you. Does that make sense to everybody? They show you the mass. They don't show you the protons, but you can find the protons easily, and you know that helium always has protons, 2 protons. Okay. Alpha decay is actually not that common. Usually beta decay is a little bit more common, so I would probably get more accustomed with beta decay, but sometimes just learn both of them. They're fairly simple, I think, if you know the rules. Cool. You had a question?
I lowered my hand, sorry.
No problem. All right. I take a little bit for this one. This one's a little bit more thinking. This one is where you really should be making conclusions, making inferences, and doing something like that. This type of question. So take a minute for this one. Okay. Let's go over this. All right, so the correct answer is actually A. And a couple things you have to note before we even start the question. Researchers observe an increase in acetyl-CoA in sec deletion cells. Again, what does sec deletion cells mean? Sec deletion means higher levels of phospholipids, right? So why is there more acetyl-CoA why do you increase levels of acetyl-CoA when you increase the amount of phospholipids? Next thing, well, what are phospholipids? As the name implies, a phospholipid is a lipid, it's fat. What does our body do with fats? Well, we went over this a couple weeks ago. Fats get turned into fatty acids, mostly lipids. When they get metabolized, they get turned into fatty acids. Fatty acids then get converted into acetyl-CoA through a process called beta oxidation. And then from the acetyl-CoA, you have a lot of acetyl-CoA per fatty acid actually when you do beta oxidation. And then that acetyl-CoA goes into the Krebs cycle and it just spins and spins and spins, makes a lot of energy, right? That's why fats are really good for giving you a lot of energy, because you can make a lot of acetyl-CoA, and because you have a lot of acetyl-CoA, you can make a lot of cycles of the Krebs cycle, okay? Now, let's move on to the actual question. A, SEC deficient cells produce higher levels of phospholipids, which are converted into acetyl-CoA through beta oxidation. That's basically exactly what I just said, so that seems like a good answer. B, SEC deficient cells must decrease flux through glycolysis in order to conserve energy. Lipids and glycolysis are not related pretty much at all. A little bit you can have control over it because obviously it happens in your body at the same place. If you're digesting a lot of fats, you don't also want to be metabolizing a lot of glucose at the same time. So if you're digesting one, you don't want to digest the other, or metabolize one, you don't want to metabolize the other, otherwise you make too much energy. But they're really not really connected, and actually having a lot of phospholipids, you would not need to conserve energy. You would actually want to kind of get rid of energy because you have too much of it. So this one doesn't really make sense. Acetyl-CoA accumulates as the Krebs cycle stops. Krebs cycle wouldn't really be stopping because you have a lot of energy. If you had energy deficiency, then the Krebs cycle would stop. If you didn't have a lot of lipids, if you didn't have a lot of carbohydrates, then the Krebs cycle would stop because you have nothing to fuel it, to power it. Or if you got rid of one of the intermediates or enzymes in the Krebs cycle, it would also stop. But didn't really mention that you're actually having a lot of fats. Eventually, maybe Krebs cycle will stop because your body will say, I have too much energy, I don't need to make more energy. And it will kind of like put the Krebs cycle on hold. But that doesn't really explain why the amount of acetyl-CoA is increased, right? Because not directly, at least, right? It's acetyl-CoA increases because you have beta oxidation, which then leads to a lot of Krebs cycle, which then leads to negative feedback loop, which leads to less Krebs cycle, right? Which leads to accumulation of acetyl-CoA, but that's like 7 steps away when you have the answer just like 2 steps in front of you. So it wouldn't be C. Hopefully that explanation made sense. And D, cells with SEC59 can only use phospholipids for energy production, causing beta oxidation to occur more rapidly. This one doesn't really make sense at all. Most cells don't only rely on phospholipids for energy production. A lot of them rely on glucose. So that's first thing that's wrong. But just pretend that they were, even then, that wouldn't explain why getting rid of SEC59 and increasing the amount of phospholipids would cause beta oxidation to, like, it doesn't really make sense, right? So it wouldn't really be D. The most, again, sometimes like the simplest answer, the most straightforward answer is the correct one. Like in this case, you just have to look 1 step in front of you. As long as you know phospholipids are lipids, get turned into fatty acids, made into beta oxidation, and you could really get A, right? One of the nice things about the MCAT, it's multiple choice, so you didn't have to even think of this answer if you didn't need to. They give you the 4 answer choices, and you just have to find which answer choice makes most sense, right? So they even, like, as soon as you see A, you might have not thought of it before, but as soon as you see A, you're like, oh, that makes sense. That must be the right answer, okay? And out of these, this is the one that makes the most sense. Any questions before I move on? I believe we have one last question.
Or can I share quickly?
Of course,
So
please.
I'm noticing with the elimination, because you said we could get to the answers by just eliminating, and I'm noticing that you eliminate some choices based on the fact that they're not really answering the question at hand.
Yeah, exactly. And
Safe to do that.
sometimes elimination,
Mhm.
yeah, sometimes elimination is easier. Sometimes just knowing the answer is easier, right? If you're sure something is wrong. I always, because I usually had a lot of time, especially for the biochem section, I would, even if I knew like A was the right answer, I would still go through B and C and D and just make sure that they were wrong, just because I didn't want to like be wrong. But sometimes you want to get the answer initially just because you know it's right or because you know everything else is wrong. As for what you said about eliminating answer choices that don't answer the question, of course, yeah. Like for this one, which conclusion, like some of these don't really even like they're like too complex, right? They don't really get to the actual reason. Again, with this one, it's not, there's too many leaps and it's not necessary to do all of that. And to answer just a simple question, right? They're just asking you a very general, simple question.
Or the common
That makes
MCAT
sense.
taker probably wouldn't know some of these. So like when we talked about those desmosomes, so we can just eliminate those answers. Like when it goes way beyond, and like you said, some of them are just straight up ridiculous. Like we're looking at this question in 4, you know, where it's saying that it's the only, what is it? Acetylcholine only uses D. Cells with Sec59-1 can only use phospholipids. So that only was kind of an extreme, and we're not really sure because there might be other ways. You know,
Right,
obviously, we said glucose was the major way. So the point of it, we can eliminate also based on the fact that the answer choices are nonsensical or probably even, like you said, just too extreme.
Yeah, and
know what was going on.
Yeah, I agree with you. Like, and that's actually something I think I mentioned maybe a couple times before, but if not, I'll mention again. I felt confident in my review enough to the point where if I saw something on the MCAT that I like, a word that I didn't know, like for example, with this one, desmosome and hemidesmosome, I really don't know what those really mean. And probably I've heard them before, but I don't really know what they mean. And it's not mentioned anywhere in the passage. If I don't know what it means, it's not mentioned in the passage, it's probably not the right answer. That was the logic I went into the MCAT test with, because I felt that if it was important enough, I would have seen it before and I would have known it. And that's kind of like what I was going on. Now, if I don't know what desmosome and hemidesmosome mean, but I know what A and B mean, and I know A and B are wrong, then I would be like, okay, well, now it's 50-50, and I would guess. But I would, as soon as I see these, I would like kind of like ignore them at first and just try and find the answer between A and B. That's something I would recommend, especially if you feel confident that you know a lot, because these like questions are there really just to have answer choices.
Yeah, well, that one was an easy, but like that SRP one, you knew it was I think you told me it was S1R, and you knew it was a transcription factor, and I'm not sure if you got that from the passage, but you know,
which one?
That same one with the SR1, you just pointed to it. Which one? SR1P.
This one?
Yes, you knew it was some sort of transcription factor, I guess, from the passage, but for the common person, SR1 RNA for
right.
RNA.
I actually didn't. Right. So like I said, this one I actually did based off of process elimination. This is one of the cases where I was like siRNA. That kind of makes sense, but like I don't know it, so I kind of skipped it at first, and then I went to B, and I was like, B doesn't make sense. C definitely doesn't make sense. D doesn't make sense. So I was like, oh well, probably is A.
B was ridiculous. So because if you're radiating a cell, you're gonna damage the rest of the good cells. So that was gone. Plasmid, we didn't really.
Plasmid would be like the opposite of what you want to do, right? Yeah. Mm-hmm. So sometimes process elimination is helpful if you don't want, but like generally, like a lot of these are like I mentioned with the desmosome, like they're kind of like they're just, and like here, this one is another good example of they just had an answer choice because they needed a fourth choice. So like these are the three real answer choices. This one's really like going like way too like
out there.
deep and like really not necessary, right? And sometimes that's just something you pick up when you do a lot of practice questions. You'll notice like, oh well, this like instinctually this doesn't feel right. Doesn't feel like the right answer. It doesn't feel like something that they would want, right? And that comes with practice, of course. But all right. Any other questions before I move on to the last problem? If not, then here we go. Now this one kind of uses everything that we've talked about for the other four questions and kind of, or most of it, and it kind of like brings them together. So really want you guys to think a little bit about this one, but if you guys have been thinking about the last ones. How can I get access to the question on this website? Oh, you can use the link. That's how I got them. I just used the link. And I'll post it again. I'll post links to actually the Google Drive, and the Google Drive has the exact link to the questions, and then you can just navigate their site. Okay. And so the answer is actually B, right? So yeah, I know Blackstone posted it in the group chat, which is fine. It's not a problem. We're going to go over it right now. Anyway, so YEP plasmid encodes a gene for ampicillin resistance, right? And ampicillin is, you probably should know, is an antibacterial, right? Or yeah, it's just like antibacterial. Researchers treating an YEP transformed bacterial colony with ampicillin should expect what? First thing it should expect, everything. Well, first thing we want to talk about is the YEP we talked about before is linked to the overexpression of, I believe it's, what was it called again? Oh, Sec59, right. So this means overexpression of Sec59, so increased levels of Sec59, right? So what would happen, and it's also resistant to ampicillin. What happens if you had a whole colony that you treated with ampicillin, and this is actually something that's really commonly used, and this is how you get vectors in the first place. You have a whole colony of bacteria. Some of them have the gene that you want, some of them don't. Well, how do you isolate the gene that you do want, which is a Sec59 that's linked, the gene is linked to an ampicillin resistance? Well, you just put ampicillin in. Anything that doesn't have the gene you want, in this case Sec59, will die. Everything that's still alive will have Sec59, pretty much. So what we expect is that the surviving cells will all be having Sec59 or having overexpressing Sec59, whereas all of the ones that are died don't have Sec59, okay? So we're going off that assumption. All of the surviving cells are overexpressing Sec59. Okay, now let's look at the answer choices one by one. Surviving cells have lower levels of PC compared to PC levels in untransformed cells. What did we say about Sec59 overexpression and amount of phospholipids? It doesn't change it, right? We covered that in an earlier question, actually, and that's one of the reasons why I said pay attention to the other questions as well. When you're answering a question, think about the questions you already did. So we already know that Sec59 overexpression doesn't lead to decreased or increased levels of phospholipid expression, right? Stays the same as if it was normal, right? So we don't expect the surviving cells to really have any difference in terms of phospholipid amounts compared to normal cells, right? So A would not be right. Surviving cells have similar levels of P and PI compared to P of untransformed cells. Yes, we expect them to have the same levels. Some YP cells to remain sensitive to ampicillin? No, because if they were sensitive to ampicillin, they would have died. So it can't be this one, right? Whenever you treat with an antibacterial, anything that's not resistant to the antibacterial dies. That's just how it is. Acquired resistance to develop rapidly in cells with the YPP plasmid. This is kind of like a weird way to say it. Acquired resistance to develop rapidly. It's not how really it works. Either the genes have the resistance, which in this case they do, and the cells either have the resistance encoded by the gene, or they don't. And the ones that died didn't, and the ones that lived did. So it'd be D. So D wouldn't be D wouldn't be the right answer. And like I mentioned, B is the right answer, right? And we covered most of that earlier already. But with this graph, right? Phospholipid levels remain constant when you overexpress Sec59. Does that make sense? Any questions about that at all? Any questions about anything at all? Anything that we covered so far? All right. If not, then I want to thank you all for joining us for today's lecture and for just the MCAT program in general. We will still be having office hours as usual tomorrow night, but this is probably the last program we'll be, the last session of MCAT help we'll be having for at least a little bit. We are planning to come back and do some more office hour sessions, more one-on-one sessions with students by appointments. We're thinking of other things that we could do to help with MCATs that are coming up in January, but for a little bit, we're going to be taking a break. All the lectures will, of course, be posted. All the PowerPoints will be posted. Everything will still be up for you guys to look at and study on your own time. Hope you've enjoyed. If you have, and I please ask this, if you have any feedback, positive, negative, it doesn't matter. Please, on our GroupMe, we post a poll about this program. Please just fill out the poll as much as you feel comfortable. If you don't want to answer a specific question, just write not applicable. If you can't answer a specific question, write not applicable, but please just give us any feedback you can. Give us any tips, anything that you liked, anything that you didn't like. Give us some information about how you attended our lectures, whether you attended live lectures, whether you attended office hours, whether you watched the recordings on YouTube or on our website. Just so we can learn our demographics. So please go to that poll on a GroupMe, which we'll be posting again today, but it was posted a couple days ago, and fill that out if you can. Yeah.